there's a lot of description. The microbiome can be both, I think it has an enormous potential. It can also be a bit buzzy. So maybe just define what we're talking about when we talk about the microbiome in and of itself, and then we can talk about what it can do.
Sure. I just want to step all the way back, if you don't mind, and think about one of the biggest epidemics we have in the 21st century, and that is the epidemic of chronic disease. Almost every other person in the room has chronic disease of some kind, and so I just want to be super clear. Everything I'm going to say here today is about chronic disease, not about acute disease, which a lot of the physicians in the room are thinking about and doing a great job with. So before it gets to the acute stage, the chronic disease is at a sort of at a molecular biological level. It manifests itself maybe several years before it actually becomes visible to you. It's a sort of a silent thing that's going on. And so in order to address that, you need to understand some of the basic molecular processes that's going on in your biology. And if you want to understand molecular processes in your biology, you have to just not only understand what's going on in the human cells in your body, you have to understand the entire ecosystem of what's going on in the body. And a huge part of the ecosystem in the body is your microbiome. So you have, for every human cell in your body, you have about 1.6 microbial cells in your body. And when I say microbial, I mean not just bacteria, I mean viruses, fungi, archaea, all kinds of different microbes are in your body. They're in your gut, they're in your mouth, they're on your skin, they're everywhere in your body.
And this is, to be clear, non-human flora.
Non-human.
This is not part of your body. These are other creatures, other.
You're a walking ecosystem, and you're like a rainforest. There's a lot of stuff going on in your body, and that's how we evolved as humans, right? And so if you don't understand how that flora is interacting with your human cells, you just cannot understand molecular processes. So that's the basis of this entire field. Just one more point. Just think about the number of genes you have in your body, right? You have about 20-something thousand human genes in your body. Guess how many microbial genes are in your body? 100 times that. It's a few million, 2, 3, 4 million microbial genes are always doing something in your body. So if you ignore the microbial genes in your body, it's impossible for you to understand what's going on in your biology. So that's the fundamental of the microbiome. If I then go into what can we do about this, there's actually a lot of great technology available today, and I'm happy to talk about what we do. I represent a company called Viome Life Sciences, where we are bringing two key technologies. One is a molecular technology called metatranscriptomics, which is understanding the activity of not only the microbiome, but of the human cells that interact with the microbiome. That's one technology. It's a molecular technology. And the second is AI. And the reason we need AI is because the amount of data that you collect from these kinds of molecular interactions going on in your body is just so massive. I mean, I'm not talking about just identifying a few pathogens, which we love doing that. We kind of reduce everything to, oh, here is the organism that's implicated in this particular disease, so let's just kill it. And in fact, that's probably one of the reasons why we've ended up with almost a crisis in our chronic disease state, because we just took the kill the bugs philosophy so far after the Industrial Revolution, right? With the antibiotics, with all of the hygiene that we do every day, we've just basically killed a lot of the rainforests that are in our body. It's just become like a desert. And so once you have that, you're actually stopping many of the great commensal processes that are going on in your microbiome from helping you. Lots of critical things you need, anti-inflammatory things, vitamins, many different things that we analyze in our using our technology, both metatranscriptomics and AI. You are actually getting rid of it because of the modern lifestyle. So that's what we set out to address in Viome.
Before we get into AI, even if people understand they have this rainforest inside their bodies,
yeah.
How do you sample it? How do you capture it? How do you measure it?
Yeah. When you think about acute disease, of course, you always want to think about some kind of invasive, process by which you can find things in your body and so on. But it just turns out that even with non-invasive samples, you can get a really great view of what is going on in your body. So think about a saliva sample, just a little bit of spit from your mouth. I can tell you with my technology today, the number of specific organisms you have in your mouth, and all of us have a lot of organisms, hundreds of organisms. I mean, I probably have 200 something organisms in my mouth, even when I keep it really clean, because there's a lot of commensal activity going on. And by the way, it's not just about the organisms, it's about what is the function, what are they doing in your mouth. It's not, I mean, you hear about things like Akkermansia muciniphala. Akkermansia is actually a really good bug. If you think about metabolic health, it can help you, but it can also be associated with, Parkinson's disease and Alzheimer's and so on. You can think about E. coli. Everybody knows about E. coli. You think about that as a bad bug, but turns out it produces vitamins like K2, for example, that is essential for us. So the point I'm making is it's not enough to know what the bugs are. You need to know what exactly are they doing, because they can be good or they can be bad depending on the environment they're in. It's like put a good person in a bad environment, they can behave badly, right? That's kind of what's going on with the microbiome. So our metatranscriptomic technology basically looks at what is each bug doing? What are they producing in terms of the metabolites? But actually, we look at transcripts, which is the precursor not only of metabolites but of proteins, right? So it's not the DNA that's important; it's the RNA that's really important, because the DNA is static throughout your life. I mean, your DNA just stays the same from the time you're born to the time you die, but your RNA is changing constantly, and that's in fact the difference between when you're healthy and when you're sick. And so we measure the RNA of the human as well as the microbiome, and then we say what is the interaction between the two that is resulting in a disease phenotype of some kind. So that's what we do in bio.
So you collect the sample. That analysis, I assume, is aided by AI and enabled by it?
Yes, very much so. So just to, again, make this super accessible, we have a consumer product today, and we have people from more than 100 countries who purchased this product. We have hundreds of thousands of people. We have more than 500,000 samples that people have collected. This is all done at home. You purchase the kit arrives home, you send back non-invasive samples, your spit, a little tiny bit of your stool, or maybe a little finger prick blood. You can just send that in, and we can see a ton of what is going on in your body and actually measure pathways and figure out what are the right lifestyle factors that could help you. For example, what do you want to eat? What do you not want to eat? What do you want to avoid? The same food can be good for me, but bad for you. It's not about the food.
Really, you can detect that to that level of personalization.
Oh, absolutely, absolutely. We've written enough papers about this topic. So, for example, a banana can create a glycemic spike in me, but it can be totally flat for you. Bread can create a spike in you, but can be totally flat for me. Why? Because your microbiome essentially consumes the food first, and it generates a lot of things that your body then consumes. That happens a lot of the time. So we've done enough clinical studies to see what the impact is. So if you come in today into our service and you give us the samples, I can tell you what is the glycemic response in your biology for every single one of the foods in our food database, and we have hundreds of foods in our food database. We can tell you.
Peter has millions.
Peter has millions, and maybe I need to interact with him and figure out how we can get those into our system as well. In fact, that was one of the things follow-ons I wanted to. Okay. But I can also tell what is the level of immune activity going on, how much of your oxalates are being metabolized, how much sulfide gas are you producing both in your mouth and your gut. All of these things are inflammatory activities. I can measure all of those things with those non-invasive samples, and then I can send you through an app, which is actually pretty cool. You can check out our app. You can see what kinds of things to buy in your grocery store. But let's say, I tell you what are your superfoods, I tell you what are your avoid foods. But let's say, you cannot get enough superfoods in your diet. I can get you a precise, personalized set of supplements, which are in a capsule or in a stick pack with probiotics and prebiotics in your home, so you can use it once per day. And this is a subscription that you can have forever. And I now have clinical studies that shows what is the impact of these specific ingredients, because we take it so precisely for your gut, your mouth, and your metabolism, that we can see the improvements in your pathways, short chain fatty acid pathways, immune pathways, metabolic glycemic pathways, and so on and so forth. We've done enough studies now to show those improvements across a huge population, hundreds of thousands of people in the world. So that's kind of our basic service. And so just to answer your other question about AI, it's impossible to understand this level of molecular data without AI. So we collect something like, I don't know, we are in the petascale right now. It's in the quadrillions, right? So 50 plus quadrillion biological data points. There's just no way that anybody can understand molecular data at that scale without AI. So we apply AI of the supervised type because we collect phenotype data, so we can do a lot of analysis, knowing what kinds of problems people have, diagnoses, surgeries, lifestyle factors, demographics, etc. We also collect data about their, all the changes they've made in their, the drugs they take, for example. What drugs work for you and what drugs don't work for you, something I can analyze through my AI, right? So I do unsupervised learning. I do all kinds of, new self-supervised, Gen AI stuff. I mean, you can talk about this for a long time. That's my background, by the way. I've lived in the world of AI for the last 20 years, and I bring in the entire tool set of AI into this huge world of massive molecular data sets that we are collecting now. In fact, this is the largest gene expression data set in the world right now, and we are applying all the advanced techniques of AI to make sense out of it and to provide consumer level insights to anyone. I mean, I want to get this out to all 8 billion people in the world if I can.
So let's say you've got a customer, they send in their samples, you do your analysis. What are some of the chronic conditions you can help them manage, and do you have any success stories?
We have tons of success stories. I mean, we have customer testimonials that are coming in all the time. We deal with people who have, metabolic issues, pre-diabetes, diabetes, people bring those down. We have people with autoimmune conditions, arthritis type conditions. These are chronic inflammatory, low level, problems that they've had for a long time. A lot of gut health issues, right? IBS is a very, very common thing. People who have come in with skin health issues, and we are now publishing all of these results. Oral health issues, this is our latest thing with, when you look at periodontitis, which actually, it turns out caries is the most common chronic disease in the world. There's a ton of people, I don't know, it's 70% or something people have some level of caries. But even periodontitis is a close second. You can do certain things in your mouth, not only take care of it, you can have lozenges, you can do, flushes, you can do lots of things that can help you improve periodontitis, caries in your mouth. All the gut issues that I'm talking about, you can help. There's so many conditions that we've been able to address using this technology that I would just suggest you look at some of the case studies on our website.
What do you think about the, obviously a lot of stores sell probiotics, prebiotics. It's a whole industry of various combinations, and many promises are not quite made, but at least hinted at in their marketing. Like, how effective are those compared to?
Yeah, so there are two problems with what are normally sold in stores, in pharmacy and so on. First of all, they don't know anything about you. They're trying to sell you something. So if it's not personalized, it means nothing in my mind. So that's the problem number one. Problem number two is, there's just, standardized sort of, claims that are being made. This will help you with digestion. This will help you with, abdominal pain. Whatever things. Those are to be studied in particular populations, and then you can say what the impact is. So those are two things we do. First, we understand what's going on in your gut, and we only give you, for example, let me give you an example. If we see short chain fatty acids are very low in your gut, which is anti-inflammatory in nature, so you want it to be low as high as possible, short chain fatty acid production in your gut. If you see that's low, we have multiple choices. We can give you probiotics that are fundamentally short chain fatty acid producers, right? If we see that you don't have such organisms in your gut, then we could give you a probiotic. But let's say you have such organisms in your gut and we see it, but they're not active. If they're not active, then we can activate them with a prebiotic and say, hey, look, give them the food, activate them, let them produce the short chain fatty acids, right? If that is not possible, then maybe we can give you the butyrate or the propionate or one of the short chain fatty acids directly, right? So there's so many options available. So if you understand the biology, then you can decide what is the right thing to do. So if you just walk into CVS and just buy a pack of probiotics or something like that, not very likely that it's going to make a difference to you because it doesn't know what is going on in your body.
All right, I'm going to tell my mother-in-law when I get home.
Yes.
Do we have any questions for Guru up here? She's got a mic right there.
So the omics is a big field. There's lots of different types of omics. So I'm just wondering, how does looking at the microbiome sort of in isolation from other things that are going on, how do you get the complete picture? And do you miss anything if you're not capturing all the different omics that might be going on with someone?
Okay. So first of all, I don't want you to walk away thinking that we only do microbiome, even though that's the title of the session. It's actually metatranscriptomics, which means we look at the human transcripts, we look at the microbial transcripts, and we look at the interactions between the human and the microbial transcripts in order to decide what is likely to happen, what is happening today. So that's point number one. Point number two is, why transcripts? Why not DNA? DNA is a static thing. It doesn't change at all, right? So when you get diabetes or when you get an autoimmune disorder, your DNA hasn't changed. What has changed is the downstream gene expression. So you can measure RNA, which is a gene expression, or you can measure proteins, but proteins are super dynamic. There's hundreds of thousands of proteins in your body, and it's very, very dynamic. It just lives for a certain period of time, it just goes away. So it's very difficult to do proteomics. And if you want to do metabolomics, it's even harder. It's small molecules, and they're very, very targeted panels. They don't have large-scale panels for it. So we decided very early on that RNA, meaning transcriptomics, is the right point in the sort of the biological dogma to focus on, because it gives us the precursor to disease, and it also is an advanced enough technology that we have developed over the last 10 years, a way to make it cheap and accessible and make it a consumer product. So for $100 odd, that's the starting point for our test today. You can go into our website and buy it right now if you want. If you want an advanced service, you can get $200, $300 service as well. But we get a ton of data about what is going on in your dynamic biology with transcriptomics. We think that is the right point at which to look at the biology, and we are doing clinical studies to show that that is the case. There was another question here.
Oh, let's go that way, and then we'll come back.
All right.
Hello, I'm Erin. I'm a health psychologist. I was just wondering, do you have any published documentation on epigenetics?
So, epigenetics is one of those sort of, I think, all-encompassing terms that includes anything that is not super well understood right now, kind of thing. I mean, that's just a little bit of a skeptical view of epigenetics. But to be more clear, I would claim that transcriptomics, metatranscriptomics, is part of epigenomics, because it's actually, there's many different processes that are going on that are converting your genome into active biological molecules, and the transcripts play a huge role. The signaling transcripts, the translational part of the transcriptomes, all of those things are playing a huge role. So that is epigenomics. At the same time, there are some other aspects of epigenomics, like, for example, methylation factors and so on, right? Those are not directly measurable by transcriptomics. However, there are precursors to methylation that can be detected even in metatranscriptomics that we do. So yes, we do have a number of publications on our website. Please go into our science section, go into the publications. We have several, I don't know, more than maybe about 20 publications so far, talking about everything from the molecular science to the clinical applications that we are showing. And to be honest with you, I don't have I haven't had the time to publish everything. we are generating from a data insights perspective, but I promise you that we'll publish more.
And I promise one last question up front.
Have you segmented the data to compare related people? And if so, what were the insights?
Interesting question. So we have not done enough to talk about, for example, what are the similarities in the microbiome among people within the same family, for example, right? We will probably do that kind of analysis shortly, and maybe we'll publish. I mean, I write blogs every month, so happy for you to take a look at it, and this can be a great blog topic. The one thing that we notice is that there are certain commonalities among people who live in the same household because of their lifestyle factors and the environment that they live in. So we had there was an intern who did a little bit of work in that area, so we saw some similarity, but we haven't scaled it to the entire population and done that analysis. So I'll take it back as an analysis that we can write a blog about maybe in the next few months.